Good morning, students. So today we are going to explore one of the crucial hormone in our body or human body is known as insulin. Insulin plays a vital role in a glucose homeostasis. As we know, insulin is produced by the pancreas. Insulin release is a finely regulated process which helps our body to utilize a glucose for energy utilization. By understanding today's topic, insulin release, it will not only deepens our knowledge about human physiology, but also appreciate the pathophysiology of diabetes mellitus. So, in today's topic, we are going to I'm going to explain you about the how does insulin releases in our body. So, let's consider a person who have taken the meal can be breakfast, lunch, or dinner. So, as we know, our normal blood glucose level is considered to be 80 to 100, between 80 to 100 mg per deciliter. After taking the meal, there is an increase in the blood glucose level. So, what does body does to this increase the blood glucose level? If the body's blood glucose level goes beyond 70 mg per deciliter, the body transport this excessive blood glucose to the pancreas beta cells. So let's consider this blood glucose level is entering to the beta cells of the pancreas. So glucose is uh, entering into the beta cell. So glucose is entering into the beta cell via the transport known as a glucose transport. Glucose transport is the channel or medium through which glucose is entering into the beta cell. After entering into the glucose, this glucose gets phosphorylated. Glucose is converted into glucose 6 phosphate. During this process, conversion of glucose to glucose 6 phosphate, one molecule of ATP is released, and this ATP molecule works on the another transporter which is present in the beta cells of the pancreas known as a ATP sensitive potassium channel. So in a normal condition, there is an influx of the potassium channel inside the beta cells of the pancreas. But now, as we know, during this glucose conversion of glucose to glucose 6 phosphate, one ATP molecule is released. This ATP molecule works on this potassium channel and blocks this potassium channel. By blocking this potassium channel, there is a dysregulation or influx dysregulation of potassium channel. So this beta cell gets depolarization. So as this point, the beta cell is depolarized and there is a sudden increase in the influx of the calcium channel. What is the significance of this calcium channel? Influx of the calcium channel. This calcium channel helps insulin which is present in the beta cell in the store format. This calcium channel helps insulin to get with the help of calcium channel, insulin is released inside the blood. So with this master diagram, we now know how does insulin released inside the blood in the blood. So let's see by this master flowchart. As we know, glucose is the key regulator of insulin secretion from the beta cell. As we know, if our blood glucose level goes beyond 70 mg per deciliter, it's transported to beta cells of the pancreas, and glucose is entered into the beta cell via another transport known as a glucose transport. Glucose transport is the medium or channel through which glucose is entered in the beta cell. Now, as we know, glucose is entered in the beta cell. Glucose get phosphorylated and converted to glucose 6 phosphate. And this conversion of glucose to glucose 6 phosphate, one molecule of ATP is generated. And this one generation of ATP molecule inhibit the ATP sensitive potassium channel by blocking or dysregulating this ATP sensitive potassium channel. They inhibit the influx of the potassium channel, and by doing the or stopping the influx of the potassium channel, membrane gets depolarization. The membrane of the beta cell gets depolarized. So as we know, now the membrane of beta cell is depolarized. There is a sudden influx of the calcium channel inside the beta cell, and this calcium channel helps the insulin to get released inside the blood. So this is the overall process of insulin release in the blood. So the next today's next topic is the insulin action. So after insulin is released inside the blood, the uh, insulin is transported to three target organs. The target organs, there are three target organs for the insulin: the liver, the adipose tissue, and the skeletal muscle. The target organs is the where glucose is stored in fat and glycogen. So these are the three organs of insulin where insulin acts. So what does insulin acts on this? How does insulin acts on this target organ? So there is a suppose this is the mm, uh, membrane of the three target organs. So as we know, insulin is transported to the three target organs, and insulin is attached to the tetrameric receptors. So this is the tetrameric receptor of insulin. So this insulin is gets attached to this tetrameric receptor, and it activates the IRS complexes, and this IRS complex perform the process of trafficking and docking. What is the trafficking and docking? There is another transport which is present in the vesicles inside these target organs, known as a glutatransport. So GLUT4 transport, by trafficking and docking process, this GLUT4 transport is transported to the membrane of these three target organs. Now, GLUT4, which is before previously present in the vesicles of the cytoplasm, now this GLUT4 transport is transported to the cell membrane or cell wall of these three target organs. Now, as we know, GLUT4 transport is present in the cell membrane of the target organs. Now, glucose can enter through these target organs. What is the significance of GLUT4 transport? For the storage of glucose, they need a medium or transport for the storage of the glucose. So, with the help of this GLUT4 transport, glucose now can enter or can be stored in these three target organs. So, now we have covered both two processes: insulin release and uh, insulin action. So let's gets back into the main today's topic, which is diabetes mellitus. As we know, uh, diabetes may be defined as diabetes is a chronic metabolic disorder characterized by elevated level of glucose in the blood. This occurs either because the body does not produce enough insulin, or because the body cells do not respond properly to the insulin. So there are two types of diabetes mellitus. Type 1 diabetes, where body's own cells, type 1 diabetes is an autoimmune disease where body's own cell destroys the beta cells present in the pancreas. So the beta cells of the pancreas gets destroyed. So as we know, beta cells produces insulin, so there is no insulin, hence the blood glucose level gets increased, and which cause diabetes mellitus. And type 2 diabetes is where body does not respond well to the insulin. So let's see the pathophysiology of type 1 diabetes, where body cannot make enough insulin. Type 1 diabetes is an autoimmune disease in which islet destruction is caused primarily by immune effector cells or cytotoxic T cells. As we know, there are two types of T cells: helper T cells and helper T cells and T cells. In the type 1 diabetes mellitus, this helper T cells works against our own beta cells of the pancreas. Type 1 effector reacting as endogenous beta cells antigens. So this is the overall diagram of the pancreas. As we say, there are islets and beta cells and delta cells. Sometimes this cytotoxic gets mad and they kills body's own cells.